Exercise 7 is all about understanding what the environmental conditions need to be in order to grow up an organism successfully. So we are going to be varying osmotic pressure, temperature, and oxygen requirement of organisms, but there's lots of other things that we could test. For instance, we could vary the pH and see how that affected growth of several different organisms. So when we understand these things, like I said, it enables us to have the right conditions to grow cells in the lab, or maybe we want to kill them. And so, we manipulate the temperature and so that we're successful doing that. All right, so 3 parts to this lab. You can do them in any order that you want. Let's talk first about osmotic pressure and do a little bit review of osmosis. Okay, so osmotic pressure, remember, this is not the same thing as barometric pressure. We're not talking about air pressure. There are organisms that grow best at the bottom of the ocean because they need that barometric pressure. We're talking about how badly water wants to leave the cell here. So we're going to define osmotic pressure as how badly water wants to leave the cell. And remember, this is going to be contingent on the solute concentration outside of the cell. All right, so little osmosis review. You've probably had this 6 different times in 6 different classes. But say you have a cell that, remember, has a semi-permeable membrane around it. Now remember, that's different than the cell wall. The cell wall is dumb. It lets most things in or out. It's the membrane that's kind of the gatekeeper there. If you have a cell, but of course our bacterial cells almost always have a cell wall as well, and we'll talk about how that impacts things. But say you have a cell with a semi-permeable membrane in a hypertonic medium. Okay, so that means high solute concentration. And when I put something in these brackets, that means concentration. So that means there's a higher solute concentration outside of the cell than inside of the cell. So if the solute concentration is high, then the water concentration must be lower, and water is going to try to equalize it. So water is going to move where there's higher concentration of water to where there's less concentration of water. Okay. Somebody told me once, water follows stuff. Okay, so the water is going to leave the cell in order to try to equalize the water concentration. Okay, this is this is a bad situation for most cells to be in because the cell is going to dehydrate. Okay, so when we're talking about high osmotic pressure, we're talking about a cell that's in increasingly hypertonic mediums. Okay, compare that to a cell that's in a hypotonic medium. And that means the solute concentration is lower outside of the cell than it is inside the cell. So it's got a higher water concentration. If water concentration outside of the cell is higher than inside, then, oops, I didn't label this, water is going to be entering the cell. Okay, so here the cell may swell a little bit, but remember, because bacteria have this nice cell wall for protection, bacteria cells are not likely to burst. It would take an enormous change in gradient there for that to happen. Now our cells, animal cells, they just, they're going to pop right away. Okay, this is one of the reasons it's so important for bacterial cells to have an intact cell wall. And there's lots of antibiotics that kind of poke holes in the cell wall and leaves it open to osmotic stresses. Okay, plant cells as well. They like a little hypotonic medium to keep up their turgor pressure. And of course, isotonic is when you have no net movement of water in and out of the cell. Okay, I think where students tend to get goofed up is, remember, these adjectives, hypertonic, hypotonic, isotonic, they're relative terms. Like, you would never say this medium is always hypertonic. Well, it's always relative to something else. Okay, and keep in mind, we usually use these terms to describe the medium that the cell is in, because the medium that the cell is in is likely, the chemistry of that is likely to change much more quickly than actual cell chemistry. So you don't ever want to say, well, the cell is hypertonic. That's just not the way that's going to goof you up. Okay, so use these terms to describe the medium. Okay. There are, like I said, most bacteria cells are going to either prefer slightly hypotonic or isotonic conditions. However, there are organisms that actually prefer to live in hypertonic mediums with high osmotic pressure, and we call those organisms osmophiles. Okay, so they grow best in high osmotic pressure. Okay, and if you want to get even more specific, a halophile is a type of osmophile that specifically grows best in a little higher concentration of salt. Okay, so an osmophile, it could be any solute, it could be sugar or something else. A halophile, again, needs, not only prefers, but really needs that high concentration of salt in order to grow well. Okay, and again, that's unusual because most of the time cells dehydrate when they're in, a certain concentration of salt. All right, so what you're actually going to be doing in lab, you are going to inoculate a plate. We call these quad plates. And they actually have a different medium poured in each quadrant. They're a real pain in the neck for our lab helpers to make up. Okay, one quadrant has, I better check and get this right. Okay, because we've changed this over the years. One quadrant has 5% salt, one has 10, one has 15%, and one has 20%. I believe your data table may say 25%, but I think it's 20. All right, so increasingly hypertonic concentrations in each quadrant. If we were to really do this experiment best or control it, we would have an area of 1% salt, which is pretty close to physiological salt concentration. So we'd have a more isotonic to have a positive control, just to make sure that our organisms are viable. But what you're going to do, you're going to take your assigned organism, whatever that organism is, whether it's Bacillus subtilis or Staph aureus or whatever you've been assigned to. You should have some either broth or solid plate cultures there. And you're going to, with your loop, get some organism and then streak your assigned organism on the quadrant like this. Okay, go ahead and flame your loop after you streak it. Let it cool a little bit, dip back into your stock culture, and then streak your next quadrant. Try to streak it exactly the same as you did the first. So whether you start in the middle and move your way out, or start out and move your Okay, testing the temperature requirement is a little more straightforward. Okay, you are going to get 4 broth test tubes, and you're going to be inoculating them. Remember, when you label your broth tubes, you're going to want to put your initials, your section number, and the organism that you're assigned to. So say I'm assigned to Pseudomonas aeruginosa, just I can just abbreviate that, that's fine. And you're also going to want to fit on those test tubes the temperature that you're going to be incubating them. Okay, so let's say this is my 55 degrees Celsius tube. All right, so typically when we incubate our organisms, we throw them all in the same rack, but this time you're going to have to pay attention and make sure one tube goes in the 4-degree rack, one in the 22-degree rack, one in our big walk-in incubator, 35, and again this one at 55 degrees Celsius. All right, so you're just going to inoculate these with your loop, stir it around like you have before, and we're going to incubate them at the correct temperature and then see which ones grow. And in order to see the growth, sometimes we have to kind of finger tap those tubes a little bit to try to see if there's anything swirling around in there. The 4 degrees Celsius will go in the refrigerator. 22, this is just room temperature, so we'll leave those out. Again, 35 at the walk-in, that's pretty close to body temperature. 55, that is 131 degrees Fahrenheit for reference there, so that's pretty warm. We have a small incubator in the other lab that we keep nice and hot, and we'll see if anything grows there. Like always, we have a little terminology to describe where organisms grow the best. Okay, if they are a psychrophile, that means they are cold-loving. And you see this defined with different temperatures at different resources. We're going to say it grows best below about 15 degrees Celsius. Okay, so that's 59 degrees Fahrenheit. That's really not that cold. Extreme psychrophiles would be the ones that maybe grow only on the polar ice caps or, the Arctic Sea, places like that. But psychrophile, cold-loving. Again, we are not inoculating any psychrophiles. We have gotten them to grow, but it takes weeks and weeks, even at the temperature they like. So we're not going to mess around with those. Mesophiles, okay, they grow best anywhere from about room temperature, about 20, to about body temperature. All right, so we're going to be mostly working with mesophiles, okay, because those are the organisms that are potentially disease-causing. Of course, we need another term, thermophiles, for organisms that are heat-loving. You understand pretty soon that bacteria are ubiquitous. They live everywhere that we can think of, the whole earth over. Thermophiles require temperatures, we'll say grow best, greater than 45 degrees Celsius. Okay, so that's about 113 degrees Fahrenheit. So that's hotter than a hot tub. Okay, so you're not going to find those every day. Those are hot springs organisms, Yellowstone. They have found thermophiles growing in water heaters in homes. Okay, so, but again, they're not going to be pathogenic because that's above the temperature of our bodies. All right. So again, pay attention to your assigned organism for this part. It will be different than the first part of lab, and just inoculate those 4 tubes and get them in the right rack. All right, lastly, we want to explore the oxygen requirement of organisms. And I don't want to go too deeply into this in lab. You may talk about it in lecture, but it's not atmospheric oxygen. It's not O2 that inhibits the growth of some organisms, but it's more reactive kinds of oxygen like superoxide that has not enough electrons to fill up its outer shell, so it's always grabbing electrons from other molecules. Okay, if organisms don't have enzymes to counteract these toxic oxygen forms, that's when they can't, that's when they can't survive. So even during aerobic respiration, there's lots of superoxide that's formed. So Again, organisms that respire aerobically have enzymes to detoxify those. All right, we're going to be inoculating a kind of media called thioglycolate media. And let's look a little bit closer as to what we find in this media. Okay, obviously it has thioglycolate in it. What's that? This is a reducing agent. If we're going to test to see whether organisms can survive in the presence of oxygen, then we need to have a way to get rid of that oxygen. Okay, so the thioglycolate takes the oxygen and basically adds some protons and electrons to it and reduces it to water. There. Okay, so the oxygen, again, it's adding electrons to the oxygen, so this oxygen is reduced. So we call it a reducing agent. All right, so here's a picture of an inoculated thioglycolate tube. Before you inoculate it, these are little larger tubes, and it has a layer of, whoops, that's a little bit longer than I meant to put it, but it has a layer of green at the top. Okay, like that. All right. So the thioglycolate is throughout the tube. As we go deeper and deeper, there's going to be less oxygen. Okay, but essentially this top portion where there's green, that's essentially pretty close to atmospheric oxygen, because some is always going to be diffusing underneath the cap. We don't have the cap on super tight. Okay, so really from here on down, less and less oxygen, and essentially very little oxygen at the bottom of the tube here. What is that green layer? There's an oxygen indicator in there called methylene blue. Okay, it's going to turn green in the presence of oxygen. Okay, so we can tell which layer of the media has been oxygenated. So remember, again, the methylene blue is throughout the tube. It's only green at the top because that's the only place where there's a certain concentration of oxygen in there. Okay, also we have a small percentage of agar in here. So it's not enough agar to make it a solid media, but it's enough to thicken up the media a little bit, because we want to see where the organisms are growing within this column here. We don't want them automatically to just settle out to the bottom. So this is just a little bit of a thickening agent. Okay, and then also there's all the nutrients that organisms to use for growth, yeast extract, or sugars, salts, etc., all that other stuff. Okay, so when you inoculate this, let me just draw it again here. You're going to use your loop and just stick it down in there. Swirl it around kind of gently. We really don't want to disrupt this green layer if we can help it. So even when you're carrying it back to your bench, be careful not to jiggle it around, or certainly don't turn it sideways if you can help it. Okay, and then after we incubate, again, usually overnight, and then we keep them in the fridge till you come in again, then we're going to take a look and see where is their growth. Okay, and then we can hopefully categorize your organism based on where we see growth. So if we have growth, there's the green layer. If we have growth only in that green layer, it's cloudy in the green layer, that's an organism that has to have oxygen in order to grow, and we would call it aerobic. Okay, that's pretty straightforward. If we have growth everywhere in the tube, usually we get even more in the oxygenated layer because when organisms respire aerobically, they get a lot more ATP from that and they grow more quickly. But this is an organism that's very flexible and it can grow without oxygen as well. We call that a facultative anaerobe. Or we could say facultatively anaerobic. Anaerobe. There. Sorry, kind of sloppy there. Okay, so facultative means flexible, right? Very flexible metabolism. E. coli is sort of our poster child for a facultative anaerobe. Okay, and then lastly, we could have growth only below that green layer. Okay, and that's, we're going to call an aerobe. Okay, now we could kind of parse this out into another few categories. We could call this a strict anaerobe or an aerotolerant anaerobe. In practice, that's very difficult to tell exactly from this test alone. Your book mentions a category called microaerobes. I'll mention that. We're not going to inoculate any, but those are organisms that need slightly less than atmospheric concentrations of oxygen in order to grow, so they're going to grow kind of just below that green layer. But we're going to try to stick to these three major categories here. Okay, this picture back here, this is a nice picture of an anaerobe. You can see how we've got nice growth. It's really cloudy, the whole bottom of the tube, and then up here, that layer that was green is totally clear now. Okay, so no growth in that green layer. We'll call that an anaerobe. And whether it's strict or aerotolerant, well, we know it must be aerotolerant because our stock cultures are not, we haven't done anything to keep the oxygen out, but there's just so few growing in there that we won't worry about that. Okay. So again, just make sure you pay attention to what organism you're supposed to be inoculating. You might use a different organism for each of the three parts of this experiment.